screen, right?
Yes, sir.
So good evening to one and all. Today our guest speaker is Dr. Naresh from Fred Hutch, Seattle, US. He's going to lecture us on biology and clinical impact of tumor microenvironment in lymphomas. This webinar is brought to you by Mumbai Hematology Group. It is supported by RPG Life Sciences Limited and it's managed by Mice Ideas. I'm thankful to Mr. Ajit Mule, Mr. Sudhir Ambekar and their team from RPG, Mr. Rajesh, Mr. Kalpesh and their team from Mice Ideas, the Executive Committee of Mumbai Hematology Group, our guest speaker, Dr. Naresh from Seattle, all the discussants who are themselves eminent hematopathologists, new participants for sparing your Saturday evening. Our discussants this evening include Dr. Sandeep Sheth, who is a consultant surgical and hematopathologist, Zydus Hospital, Ahmedabad. Dr. Tara Roshni Paul, she is professor and head department of pathology at the prestigious Nizam Institute of Medical Sciences, Hyderabad. Dr. Udyan Kachi is pathologist, Dr. Udyan's lab, Vadodara, Gujarat. Dr. Parimal Sarda, who is consultant hematologist, hemato-oncology clinic at Vedanta, Ahmedabad. Dr. Ankur Ahuja, assistant professor, Department of Lab and Molecular Medicine, Army Hospital, R&R, New Delhi. And Dr. Sindhura Lakshmi, associate professor, Department of Pathology, staff pathologist, hematology and clinical pathology, Kasturba Medical College and Hospital, Manipal Academy of Higher Education, Manipal, Udupi. And Dr. Amrita Saraf, senior consultant, Department of Hematology at Sir Gangaram Hospital, New Delhi. A pleasure to introduce Dr. Naresh. He is hematopathologist with focus on lymphomas, bone marrow pathology, and molecular pathology. He has been in the field of hematopathology for over 30 years. He has worked, trained across 3 continents, North America, Europe, and Asia. Prior to joining Fred Hutch, he was at Hammersmith, Imperial College, London. He leads diagnostics, translational research, and training in hematopathology. He was the chair of British Lymphoma Pathology Group. As a part of his activities in BLPG and Royal College of Pathologists, he played a critical role in improving the standards of training and practice of hematopathology in the UK. There's over 250 publications, majority of which are in the field of hematopathology. In hematopathology research, his collaborations extend with investigators and institutions in US, Europe, Asia, and Africa. He is a firm believer of global medicine and science and in fostering international collaboration to promote translational research and training in hematopathology and other branches of medicine. A pleasure to invite him to deliver his lecture on biology and clinical impact of tumor microenvironment in lymphomas. Thank you very much. Over to Naresh.
Good evening, everyone. Let me share my slides. Okay. I'll be able to see my slide in the presentation mode.
Yes,
okay. Yeah, it's a real pleasure to come back to give a webinar in the course that Dr. Agarwal has been running very successfully for maybe more than a year now. And I have worked with Dr. Agarwal on many of his courses for many years before, and it's always a pleasure to speak on his educational pursuits. I picked a subject which is slightly different from my routine clinical practice, and it partly encompasses some aspects of my research interests. But at the same time, I think this subject is quite a difficult subject, and especially for a simple hematopathologist like me. And it encompasses, it requires expertise from many different areas, including immunology, image analysis, epidemiology, clinicians, and all those. So what I'm going to present today is more of a hematopathologist perspective, and in some ways it could be a much more narrow focus than the actual subject, what it demands. So in the next in the next 45 minutes or so, I will try to go through this topic along these lines. I will have a brief introduction, then I will talk about the key players in the microenvironment of lymphomas. Then we'll discuss a few slides on immune checkpoints, which probably most of you are well aware of. Then we'll look at what are the prototype situations in lymphomas where microenvironment is distinctly different. Then I will pick three classic typical types of other more common types of lymphomas where I discuss the impact of microenvironment, both on biology and on clinical aspects: follicular lymphoma, diffuse large B-cell lymphoma, and classical Hodgkin lymphoma. Then we will try to summarize how technologies have evolved over the last 20 years in the study of microenvironment and where we are heading. And then I will end with a few slides on how this will impact on disparities research in terms of how the microenvironment impacts on patient outcomes and what kind of disparities this is going to introduce. Just as an introduction here, I mean, I have put here a simple slide. And part of this slide depicts what we often see in solid tumors. That is, there is a tumor, there is an immune microenvironment, and the job of the immune microenvironment is to control and subdue and kill the tumor. So that, and the tumor itself will try and see how it can subdue the immune microenvironment, and this balance shows the heterogeneity of how patients and tumors evolve and present. But in lymphomas, it is slightly different because lymphoma itself is an immune cell as compared to most other solid tumors, which are not as immunologically active as a lymphoma cell. A lymphoma cell is an immune cell. So the way lymphomas immune microenvironment partly differs from those of solid tumors is that there are many immune cells which are actually supporting the tumor, and some of these things can be there even in the normal immune system. And the tumor cells have the ability, the lymphoma cells have the ability to induce a supportive microenvironment. And over a period of time, the tumor cell can become dependent on immune cells. And that also can be a problem, but also it opens up opportunities to break this dependency and treat patients. So just to understand what are the effectors in tumor immunity, I think there are a few innate immune cells like the antigen-presenting cells, the NK cells, the T cells. These are not dependent on the kind of antigen. And we do not know where exactly by standard B cells stand today, whether they are adaptive or whether they are native in terms of providing a response, and whether they do provide a response to tumor cells. That's also in question. We will briefly discuss this. But what is well established are the adaptive immunity. I mean, the two main players in adaptive immunity is the cytotoxic T cells and the macrophages. And their job is to actually kill the tumor cell. And the story is not that simple. So if everything went well and we had a very good immune response and patient is going to get cured purely by the immune cells, what would be an effective way that immune system can respond and maintain and kill the tumor? I mean, one is it needs presence of unique antigens by the cancer cells, and these cancer antigens should be presented properly to be recognized as foreign antigens. So even though these are antigens that are created by the host cells, in the process of becoming a cancer, they should be recognized as foreign antigens. So that's the first step. And once that happens, there has to be activation of the immune effector cells. And these immune effector cells then take on and attack the tumor cells. But they themselves may not be sufficient, so there should be a mechanism for them to expand. So expansion of immune effector cells is a key aspect. And there has to be continued presence of the active effector cells until the last lymphoma cell is lysed. So not only it has to evoke an immune response, then expand the immune response and maintain the immune cells till such time that all the lymphoma cells are killed. And beyond that, there also has to be an immunological memory, so that in case the tumor cells again arise, the same immune cells should be able to attack the recurrent lymphoma cells to prevent recurrence. So this is what an effective immune.
Recording in progress.
This is what is important for an effective immune response. So what are the components of an effective immune response and also the way the anti-tumor immune response can be modulated? Antigen presenting cells are key so that the cancer antigens are presented, and lymphoma cells being an immune cell, it itself can present as it can be an antigen presenting cell. And the key players, as I said earlier, are the cytotoxic T cells and the macrophages. But these two components are heavily modulated by other cells. There are many regulatory lymphoid cells, and also there are many other extracellular factors which can activate or suppress the immune system in terms of ligands. Then, of course, as I mentioned earlier, memory T cells are key for preventing recurrence. So within the cytotoxic T cells, there are key receptors which are important for either driving activation or giving inhibitory signals, suppressing the T cell response. The CD3 T cell receptor complex is an important activation signal, so recognition of the antigen in the right MHC context is key. Then you have co-stimulatory molecules, which are like 27, 28, 134, 137, and a few other receptors which give rise to activation signals. To balance this, there is also, along with activation, many of the receptors like PD1, CTLA4, they start getting expressed, or LAG3. These will send inhibitory signals. And this kind of balance between activation and inhibition is a key aspect of homeostasis of any immune response. Because if there is a foreign antigen, you want T cells to come, take care of the antigen. But once the antigen has been taken care of, the foreign antigen has been dealt with, you don't want the immune cells to go rampant. So the immune system has a check and balance that once the antigen is dealt with, you have the inhibitory signals which are expressed, and then the immune response quietens. So this is a normal physiological homeostatic response. But in terms of cancer response, having many inhibitory signals suppresses the immune system and allows the cancer cells to grow. So, and as you will see in later slides, cancer lymphoma cells can manipulate the system and can upregulate the inhibitory signals for their own survival. There are other extracellular components which are also key. One are the blood vessels and angiogenesis, extracellular matrix, many cytokines and chemokines which are delivered by the lymphoma cells or by the other immune cells, which can suppress the immune system, or sometimes it can stimulate the tumor cells. And there are also structures called lymphoma exosomes. These are fragments that are secreted by the lymphoma cells, which are membrane bound, and they contain key messages, and these can also be seen in circulation. And this can be seen in the microenvironment as well. So these are all key aspects of an immune response. So just to understand, two slides on immune checkpoints, which most of you are well aware of. For an immune response, here is an antigen presenting cell, and here is a cytotoxic T cell. The antigen is presented on the MHC class 1 or class 2 molecule, and then the cytotoxic T cell, the CD3 TCR response, the complex recognizes this and sends a proliferation or an activation signal. At the same time, it has got two other, it has got CD28, which is a co-stimulatory signal, which binds to B7-1 or B7-2, which gives a stimulatory signal. Or at the same time, it can express CTLA4, which CTLA4 and CD28 compete to bind to B7 or B7-1 or B7-2, and you get alternate different downstream signals. While with CD28, you get a stimulatory signal. With CTLA4, one gets an inhibitory signal resulting in T cell anergy or apoptosis. So you get two completely opposing possibilities. So in a case which is where you have T cell anergy because CTLA4 is expressed, if you were to block the CTLA with an appropriate antibody, you can upregulate the interactions between B7, 1 and 2 with CD28, making the cytotoxic T cells more responsive and more active and more effective. And the other one is much well known is the PD1, PDL1, PDL2 combination, where in the similar things, cytotoxic T cells also express, they start to express PD1, and the tumor cell can express PDL1 or PDL2. And when PDL1 and PDL1 or L2 is expressed on tumor cells, it binds to PD1 on the surface of the cytotoxic T cells, giving rise to an inhibitory signaling, which results in T cell exhaustion and decreased cytotoxic toxicity. And if you use PD1 or PDL1 antibody, you can block this interaction and make the cytotoxic T cell active again. If you were to use a PDL1 blocking antibody, it would block the PD1, and thereby it will block the PD1 receptor, and but it will only this blocked receptor will only inhibit the interaction with PDL1, but PDL2 interactions are still ongoing. But if you block if you use a PD1 blocking antibody, it will block both PDL1 and PDL2, thereby really giving a good activating signal to the cytotoxic T cell. So there are other mechanisms by which the immune evasion can occur. One of the key aspects in many lymphomas is they start downregulating or completely losing MHC class 1 or class 2 expression. Thereby, the tumor cell is not, the new antigens on the tumor cell is not recognized by the immune cells. Or the tumor cells can express PDL1 or PDL2. Sometimes it is because of the genomic changes, like you see in mediastinal large B-cell lymphoma. You have got translocations involving PDL1 or PDL2, resulting in upregulation. Whereas in other lymphomas, it may not be genomic changes. There are other changes which result in upregulation and overexpression of PDL1, PDL2. And you can also have other secretions from the lymphoma cells, which will upregulate what we call as regulatory T cells. And these regulatory T cells will suppress the rest of the immune cells, both the CD4 and CD8 cells in the microenvironment, thereby again promoting tumor growth. And tumor cell and lymphoma cells can secrete cytokines that can promote immune exhaustion and also result in immune suppression. So all these ways lymphoma cells can help themselves by suppressing the immune system and a local immune microenvironment. The suppression and regulation of stimulated cytotoxic T cells can occur through exhaustion, and this exhaustion occurs by expression or stimulating the expression of PD1, LAG3 or TIM3, and by increasing the numbers of regulatory T cells which express CD25 and FOXP3, or by bringing in recruitment of monocytes and macrophages. And these monocytes, macrophages express PDL1, PDL2, which will interact with PD1 expression on cytotoxic T cells, suppressing them. Then you also have myeloid-derived suppressive cells, and there are multiple other suppressive cytokines that lymphoma cells can secrete. So here are the different prototypes of lymphomas and their microenvironment. And the two extremes are one, what we see in Hodgkin lymphoma, where less than close to 1% of the population are actually tumor cells, and rest of them are all immune cells. And the other end of the spectrum is like what we see in a Burkitt lymphoma, where there are very few immune cells, mostly being macrophages, and hardly any T cells being there. Most other lymphomas fall somewhere in between Hodgkin lymphoma and Burkitt lymphoma, where there is a fair degree of immune microenvironment. And another example is a follicular lymphoma, where the immune microenvironment is very similar to what one would see in a normal follicle. So here, the lymphoma re-educates the normal microenvironment of the follicle and uses the tumor microenvironment for its own proliferation and survival. Just to remind you, in the microenvironment, even of a normal germinal center, is there to allow proliferation of normal germinal center cells. So it uses the normal machinery of a germinal center microenvironment for its own growth and maintenance. Whereas in Hodgkin lymphoma, there is no normal counterpart for the microenvironment of a Hodgkin lymphoma. So it's a completely new, different type of microenvironment. The lymphoma cells reprogram the reactive cells and use them for their own survival. And here is a depiction of various survival and proliferation signals using different microenvironmental cells. So there are both bacterial epitopes and autoantigens, like what can happen in follicular lymphoma or MALT lymphomas, which can directly bind to the B cell receptor, giving proliferative signals, or it can be various other cells, like the stromal cells, neutrophils, macrophages, T cells, eosinophils, mast cells, all of them interacting with different components on the tumor cells, giving rise to proliferation signal. And also lymphoma cells can secrete various factors like the hedgehog proteins, APRIL and BAF, which all form ligands for important receptors, giving rise to proliferation signals. And also, there can be direct crosstalk between different cells, which we will see many examples of. And the crosstalk can be inhibitory in their normal situation, and the lymphoma cell will find a way to circumvent this. Like here, you can see the CD58 expression of the MHC class 1 expression being downregulated on the lymphoma cell, allowing the NK cell not to bind to the lymphoma cell and not to do its job. Similarly, MHC class 2 antigens can be downregulated, so the cytotoxic T cell no longer is effective on the, and again, this is something which we discussed, the expression of PDL1 or PDL2 on lymphoma cell can make the cytotoxic T cell ineffective. And various other factors that are secreted by the lymphoma cells, like the TGF-beta and galactin, all these things can result in T cell anergy or blocking of the cytotoxic T cells, pushing the CD. Like here in this situation, what is depicted is expression of Fas ligand on the tumor cell and Fas on the cytotoxic T cell, resulting in apoptosis of the tumor cells. So in effect, what it does try to do is it suppresses the Th1 kind of response and augments the Th2 type of response and moves the macrophage response from what we call as the M1 to M2. And the M2 is the one which is pro-tumoral and M1 is anti-tumoral. And so these changes are brought about in the immune cells for its own survival. So across, the immune cells can be broadly divided into those which are pro-tumoral and those which are anti-tumoral. Pro-tumoral are neutrophils, the M2 polarized macrophages, which express CD163, and a Th2 CD4 positive T cells, and regulatory T cells. These are pro-tumoral cells. And the anti-tumoral cells are the M1 macrophages, the Th1 CD4 positive T cells, and the CD8 positive T cells. So the tumor will always try to change the microenvironment from the anti-tumoral to the pro-tumoral microenvironment. So we move on to some of the examples here and to look at how these aspects that we discussed in general affects them in particular. In a follicular lymphoma, as in a normal follicle, these are some of the key players. You have the follicular dendritic cells, which are the key antigen presenting cells. Then you have the fibroblast reticular cells, mesenchymal stromal cells, and you have tumor associated macrophages. And you have got very many different types of T cells. If you take a normal reactive germinal center cell and do a CD3 stain, you will see nearly 20% of the cells are T cells. And this can be exaggerated in sometimes both in the reactive follicle and sometimes in follicular lymphoma. More than 50% of the lymphoid cells within the follicle can actually be T cells. And they are a mix of various types of T cells. You have the follicular helper T cells, follicular regulatory cells, and the regular Tregs, FOXP3 positive Tregs, and then you have cytotoxic T lymphocytes. So follicular helper T cells are a key player in the whole response, and these are CD4 positive T cells that are usually present in the light zone of a normal germinal center, a secondary follicle. And the phenotype of this is they express CD4, they express CXCR5, PD1, ICOS, and CXCL13. Most importantly, they're negative for CD25, and they're often negative for FOXP3. These TFH cells are essential for the formation and maintenance. They contribute to B cell fitness by means, and these cells also express CD40 ligand, and CD40 is expressed on B cells. So this gives rise to a stimulatory signal, and they produce cytokines IL-4 and IL-21, which are important growth signals to the lymphoma cells. Then we have the regulatory T cells. I mean, after T cell stimulation and the CD4 positive cells, then they express CD25 and FOXP3, and they change the phenotype into a regulatory T cell. And these are recruited by the malignant B cells. And the key aspect of regulatory T cells is to suppress and inhibit CD8 positive effector activity. It also suppresses CD4 positive T cells. So its job is to regulate an immune reactions, dampen the immune reaction. Then you have two types of macrophages. As I said, one is the M1 macrophage, the inflammatory type of macrophage, which suppresses the tumor cells. And then you have the anti-inflammatory pro-tumoral macrophage, which is the M2 macrophage. And distinction of these two is quite important. Just to look at different players here depicted in this cartoon, the key player, as I said, is the TFH cell with the CD40 ligand interaction, CD28 and CD80 interaction, and the antigen being presented on the follicular lymphoma cells with the MHC class 2, and that binding to the T cell receptor on TFH cells. But over a period of time, these become follicular regulatory cells, which expresses BCL6 and FOXP3. They can also dampen the TFH cells. Then you also have the regulatory T cells which come into play, which are CD25 positive cells, and these will dampen any cytotoxic T cells. And then the various things that happen within the follicular lymphoma, the cytokines which are secreted like CCL2 and CSF1, they start recruiting monocytes and change the monocyte phenotype into AM2 macrophage. And these cells express PD-L1 and L2, which can also suppress the cytotoxic T lymphocytes. So, and if FDCs provide an important growth along with the antigen presentation, they stimulate the follicular lymphoma cells. And you also have the reticulum cells, the stromal cells, which can produce extracellular matrix, which is also pro-tumoral. With this in background, it was somewhere in the early 2000s that the first paper came out on when they were looking at gene expression of follicular lymphoma and how the gene expression can impact on patient survival. To the surprise of this paper, that more than the transcripts that were coming from the lymphoma cells, the discriminatory transcripts were actually from the background immune cells. So this paper by Sandeep Dave, when he was with Louis Stout, came out with two critical immune response signature, what he called type 1 and type 2. And type 1 was predominantly coming from T cells with some the messages coming from macrophages, and the type 2 coming from a completely different set of macrophages, which we now understand as M2. And the immune response type 1 genes, wherever in patients where immune response type 1 gene were prominent, these patients had a favorable impact on survival, whereas those patients where immune response 2 messages were prominent, these patients had a very unfavorable outcome. So this was one of the first papers which on gene expression showed that the immune signature, the signature coming from the immune cells, were important for patient outcome. Then following this, then some of the key molecules from that were picked up by, I think, so the Randy Gascoigne's group in British Columbia, who looked at the macrophages by CD163, and he looked at, but this was very different. I mean, it gave two different messages in patients who were treated with RCBP, having a high number of macrophages in blue line here resulted in a poor outcome, like what was the prediction from Sandeep Dave's paper. But in patients of follicular lymphoma who were treated with R-CHOP, not only this did not matter, but actually it reversed the outcome impact. Cases which had a low macrophage count actually did worse than those which had increased number of macrophages. So this study was in some ways quite confusing. Then again, the microenvironment can be modulated by genomic events. So these are not entirely on their own. These events are also modulated by various genomic events, like mutations in CREB-BP results in loss of MHC class antigen expression. Like similarly, the deletion of B2M, beta-2 microglobulin gene, results in loss of MHC class 1 antigen. Mutations in EZH2 and RRAGC results in shift the dependency from TFH cells to follicular dendritic cells, and so on and so forth. So genomic events alter the microenvironment aspects of follicular lymphoma. And again, this provides, this is the same thing depicted here. The CREB-BP is a loss of function mutation, which results in downregulation of MHC class 2. But all these things open up druggable targets. And EZH2 mutation, you can be treated with, because it's a gain of function mutation. A patient becomes amenable for EZH2 inhibitors. So many of these genomic events which alter the microenvironment can also be targeted using various drugs. And then, because there is an intense crosstalk between follicular lymphoma cells and other microenvironmental cells, like here the dendritic cells, the follicular dendritic cells, the TFH and regulatory T cells, monocyte macrophages, many of these things open up druggable. These are all druggable targets, the targets being the B cell receptor or a PI3 kinase delta. All these things are amenable for treatment using specific drugs. And that's the reason why we need to understand microenvironment in great detail. In diffuse large B-cell lymphoma, this again came around at the same time, the early part from the same group. It was the group of Louis Stout. Again, Andreas Rosenwald was the first author of this paper. When they were looking at what was the gene expression pattern between different diffuse large B-cell lymphoma subsets, one particular signature, which was a component of the outcome predictor score, came out, which was not from the lymphoma cells. That was what they call as a lymph node signature. These were predominantly by the non-malignant component cells. So that was the first indication that a similar thing as what is seen in follicular lymphoma is also at play in diffuse large B-cell lymphoma. Subsequent to this, the same group then went on to study immune signatures, like in the follicular lymphoma paper by Sandeep Dave. Here, Lenz's paper focuses on two types of stromal signatures, stromal signature 1 and stromal signature 2, which was relatively independent of germinal center activated B-cell types of diffuse large B-cell lymphomas. And this impacted on survival of patients. And you can see, based on stromal signature 1 and 2, they could come up with a combined stromal score, which in turn impacted on the survival predictor score. Again, within each of these signatures, there are a large number of genes. So again, in early 2002, most of these things could not be practiced outside of having an excellent lab doing microarray studies. So people started picking up single molecules from that and started testing on tissue samples. This was a study from Barts Hospital in London, where they looked at CD3 positive cells in diffuse large B-cell lymphoma. Here are two extreme examples. One of a diffuse large B-cell lymphoma with very few CD3 positive T cells, other one with a large number of CD3 positive T cells. And as one would expect, cases with a large number of CD3 positive T cells, they did better than those who had fewer CD3 positive T cells. So the outcomes were quite statistically significantly different, with cases having more CD3 positive T cells doing well as compared to those who had less CD3 positive T cells. The same paper also looked at FOXP3 positive cells, and this was a bit counterintuitive in the sense, here is a FOXP3 low case and here's the FOXP3 high diffuse large B-cell lymphoma. One would have expected those cases with a lower number of FOXP3 cells to have done better, because this would have allowed cytotoxic T cells to act. But in this paper, you will see that the cases with a higher number of FOXP3 positive cells, they did better, which is actually so. One of the problems with this study is this study did not look in the same in the same tissues how many of these were actually cytotoxic T cells. So if you have a large number of FOXP3 positive cells but had very few cytotoxic T cells, obviously those would do badly. So I think looking at these molecules in isolation may not be very useful. This is another study which looked at the immunogenicity of the lymphoma cell itself. Here your HLA-DR positive cells where are immunogenic, they tend to do better than those diffuse large B-cell lymphoma which downregulate or turn off HLA-DR, and they do badly. In the more recent times, now from the requirement of a microarray analysis has come down. Now we can do a select number of genes can be tested very robustly on a nanostring assay. And here you can see the nanostring assay has been used in this study on diffuse large B-cell lymphoma, which looked at a signature of a macrophage, an M2 kind of macrophage. And those cases which had a lower signature of the macrophage signature, they did better than those which had a high macrophage signature on the nanostring assay. And this was true both with the CHOP-treated patients and with the R-CHOP-treated patients. So the science now moved from doing an extensive gene expression analysis on a microarray to a much more focused nanostring-based assay, and which seems to be quite reproducible and many people have been doing this now. And if you look at various studies that have been published that are in this area, you see a large number of studies, some of them slightly contradictory to each other, but in general having the same direction that immune microenvironment is key to diffuse large B-cell lymphoma and in predicting outcomes. We move on to Hodgkin lymphoma. I mean, Hodgkin lymphoma, as one would imagine, having only 1% lymphoma cells and 99% microenvironmental cells is a perfect pitch for looking at how the microenvironment can interact with tumor cells. And you can see here the microenvironment has from plasma cell to eosinophil to mast cell, other B cells, fibroblasts, neutrophils, various types of T cells, CD4 positive cells, TFH cells, regulatory T cells, cytotoxic cells, all of these things having in a very cytokine-rich environment with a lot of crosstalk between the tumor cell and the microenvironmental cells happen. And if you try to put all these things together with what cells interact with what receptors on the tumor cells, there are some key pathways that come out. The JAK-STAT signaling pathway, the NF-kappa B pathway, both the canonical pathway and the alternate pathway, and many tyrosine kinase induce stimulations. And again, this, in a sense, I think the most important aspect of the Hodgkin lymphoma physiology is the TNF receptor family, composed of CD30, 40, 15, rank, and CD95. And in the microenvironment, there are cells which have the ligands for all these receptors, giving proliferative signals and survival signal to the Reed-Sternberg cells. So the role of cytokines is also important in in Hodgkin lymphoma. And two important things that are present in the Hodgkin lymphoma milieu are the Th2 type of T cells and the regulatory T cells. Reed-Sternberg cells secrete Th2 chemokines and cytokines, dragging them into the microenvironment. And this leads to infiltration of not only Th2 cells, but also of eosinophils and fibroblasts. And they also secrete cytokines which inhibit Th1 responses and induce apoptosis in Th1 cells and in CD8 cells. Then you also have cytokines released by the lymphoma cells, which leads to influx of regulatory T cells. And some of these cytokines also have an autocrine effect on the lymphoma cells. And when people started looking at how the microenvironment modulates to outcome, I think again this was from the Vancouver group, and which you can see that they looked at, they took two groups of patients. One group of patients where the treatment was successful, another group of patients where the mix of patients who had success and failures in treatment. And the hierarchical clustering, you can see that they clearly segregate set of genes which are expressed in those which were successful and in those cases where treatment was not successful. And if you rank them, and if you look at the pathways that are that are stimulated by these transcripts, the cell-mediated immune response comes at the top, at the very top of the message. And the cell-cell signaling and interaction is also key in the treatment failure group, and genes involved in IL-12 signaling and production by macrophages are also key in the treatment failure group, along with apoptosis. And one of the key messages that came out from this paper was the CD68 the macrophage infiltration within the Hodgkin lymphoma microenvironment. Those Hodgkin lymphomas which had a large number of macrophages identified here by CD68, those had very poor outcomes, whereas those which had very few macrophages, less than 5% macrophages, those did very well. And so macrophages seem to be a discriminator, and this was one of the major studies which came into 2010. And then many other people have tried to look at these questions of what immune cells in Hodgkin lymphoma can be responsible for better or poor outcomes. And like you can see here, there are various types of like mast cells, which is innate immunity. You got macrophages, like I showed you on the previous one. You have low infiltration or high infiltration Hodgkin lymphoma cells. Then you have regulatory T cells, which can be more, and so this is going to be suppressive. So these are identified by FOXP3. And here are granzyme B positive cytotoxic T cells, showing you an activated T cell phenotype. There are some Hodgkin lymphomas which have a lot of granzyme B positive cells, and some which have very low granzyme B positive cells. And when each of these factors were looked how they impact on outcome, you can see that one here in blue is the one which are FOXP3 positive infiltrate. If there are more number of FOXP3 positive cells with that immune signature, those tend to do badly. And those with an active signature in red here, those are the ones with a large number of granzyme B positive cytotoxic T cells, they tend to do well. And just using the regulatory T lymphocytes, the low and high, dividing them, you can see that the cases with a higher number of regulatory T cells, they do worse than those cases who had fewer regulatory T cells. More recently, this year, we have had a publication from the German Hodgkin lymphoma study group, where they did a whole slide imaging and they quantitated different cellular elements within the microenvironment. Those cases who had a lower number of normal B cells, they did worse than those cases which had a high number of normal B cells. So the question comes here: what are these normal B cells doing in the Hodgkin lymphoma microenvironment? I don't think we understand this well at the moment. But what one has to be careful with these kind of studies is that there is a difference in the number of normal B cells between different histologic types of classical Hodgkin lymphoma. Like if you look at lymphocyte-rich classic Hodgkin lymphoma, they have a large number of small B cells. So whether the number of B cells can be independent of the type of Hodgkin lymphoma, that comes into question. So this is the first paper that has come out with this kind of a message, and it needs to be seen whether this can be reproduced. Now things are moving on into looking at a single cell transcriptomic analysis. You can take all the cells in Hodgkin lymphoma and type the cells in great detail, and you will see that there are various types of lymphoid cells, plasma cells, B cells, and T cells are present within the microenvironment of Hodgkin lymphoma. And these cells are very different from what one can see in a reactive lymph node. Here, this was a study looking at the T cell subsets in Hodgkin lymphoma, classical Hodgkin lymphoma, and reactive lymph node. And within the microenvironment of Hodgkin lymphoma, this identified very many different types of subsets of T cells and B cells. But this was not a study which looked at outcomes. and basically looking at differences between different types of Hodgkin lymphoma and between reactive lymph node and Hodgkin lymphoma. And you can see here in red are the Hodgkin lymphoma samples, and in blue are the reactive lymph node samples. At one end of the spectrum, you have regulatory T cells, which are markedly overrepresented in Hodgkin lymphoma samples, and you have the CD8 memory T cells markedly overrepresented in reactive lymph node samples, and rest of them falling in between. And when you look at differences, you can see that in the reactive, this one is reactive lymph node as compared to Hodgkin lymphoma, you have a significantly higher number of Th1 as compared to in Hodgkin lymphoma as compared to reactive lymph nodes. And more importantly, Th17 seems to be a big discriminator between Hodgkin lymphoma and reactive lymph node. But when you look at within the subsets of Hodgkin lymphoma here between mixed celularity and nodular sclerosis, I think again Th1 is more common in mixed celularity, but not so high in nodular sclerosis. And again, Th17 is predominantly seen in nodular sclerosis Hodgkin, but not so in mixed celularity Hodgkins. So what this comes up is, finally, the message of this is mixed celularity Hodgkin, the microenvironment is that of Th1, and in nodular sclerosis Hodgkin, the microenvironment is predominantly of Th17. And when you look at the regulatory T cells can express either FOXP3 or LAG3. In Hodgkin lymphoma, in most other situations, FOXP3 positive regulatory T cells are far more, and most of them express FOXP3, whereas in Hodgkin lymphoma, the number of cells expressing LAG3 are far more than those which are expressing. The level of expression of LAG3 is far more than the level of expression of FOXP3. And when you look at LAG3 expression, and again, the Hodgkin cells are heterogeneous with respect to expression of MHC class 2. Those cases which are MHC class 2 positive, they show a lower expression of LAG3 as against cases of Hodgkin lymphoma, which are MHC class 2 expression negative. Those have a very high expression of LAG3. And here are the photographs which show you MHC class 2 positive cells, the predominant regulatory T cells of FOXP3 positive cells, and sort of more evenly distributed in the microenvironment. Whereas those which are MHC class 2 negative in the lymphoma cells, they show a intimate rosetting by LAG3 positive cells and very few FOXP3 positive cells. So within Hodgkin lymphoma, you can it seems there seems to be a dichotomy of MHC class 2 expression and of the microenvironment. The ones which the one type of cells where you have no MHC class 1 class 2 expression, and those having a high number of LAG3 positive cells and fewer FOXP3 positive cells, and those which have MHC class 2 expression having a higher number of FOXP3 positive cells and fewer LAG3 positive cells. So we just move on to what are the current tools that are available to assess different the microenvironment. One is flow cytometry, which has been there for a long time, but the one drawback of flow cytometry is that we are not looking at the immune cells in their architectural context, and that is the limitation of flow cytometry, though we can do extensive work. Then you have a mass spec, CyTOF we can use, again, a similar problem as flow cytometry, that you are altering this architecture. Then you have immunohistochemistry. I showed you several examples. Often you can do 1 or 2 immunostains with immunohistochemistry traditionally, but now we can go up to 6 or 7 antigens we can do on the same slide with multiplexing immunohistochemistry. And fluorescence microscopy is sort of similar to immunohistochemistry. Studying 3 to 5 antigens is probably 3 to 4 antigens is probably quite easy in the routine fluorescence microscope. But now we are moving, there are so many messages to be studied at the same time. So we are moving into bulk transcriptomics. As I showed you several studies which looked at gene expression studies using microarray analysis. But this requires a lab which is heavily invested on gene expression. And there are very few labs, and it is not practical to do it on a clinical case. But nanostring allows us to do it on a single case. But again, both this kind of transcriptomic analysis does not allow in situ analysis. So you are extracting the RNA and studying. So the drawback of these studies is still that it is not in situ and the architectural context is lost. Then you also have single cell transcriptomics. You can study in much more detail single cells. I showed you one example in Hodgkin lymphoma. But again, the drawback being losing the architectural context. We can also look at immune repertoire profiling, and the repertoire of the T cell receptor, giving some idea about the neoantigen for which it is responding to. So these are different conventional tools that are available. But now we are moving on to the next generation of tools that are currently available. In our own laboratory, we are now standardizing what's called a codex system. With this system, we can go up to 40 or more than 40 antibodies. We have come up to close to 20 antibodies, which we can assess on a single paraffin section. In contrast to the conventional antibody detection system, this uses DNA barcodes and fluorescent DNTP analogs, and this is an index in situ procedure. Just to give you a brief idea about what this technique involves is that you have an antibody, and the antibody is, you conjugate that antibody with a barcode, and then you have a reporter based on DNA homology. You have a reporter, and at any given cycle, you only put 3 antibodies. So you take the section, you add 3 antibodies which are attached to the barcode, and then you put the reporters for those 3 antibodies. Then you scan the slide, you get the message of the 3 antibodies with 3 fluorescent dyes. Because there are only 3 fluorescent dyes, you don't have the problem of compensation like you have on flow cytometry. Then you remove these reporters, the antibodies will still remain. Then you add the next 3 antibodies. So you can go on and on adding new antibodies, new cycles, with each cycle adding 3 new antibodies. So you can easily go beyond 40 antibodies. And then finally, you'll be able to evaluate simultaneously, you can evaluate the expression of 40 plus antibodies. And these can also be evaluated like you would evaluate a flow cytometry plot. These messages can be taken onto a 2D or a 3D plot, and then you can ask the unique populations identified can be thrown back onto the slide to identify where exactly those cells are. And I mean, these are all, I mean, I'm not showing any of our own work because those are not published yet. These are all from the company website, which are available freely. So this is a technology which I think is going to, which has already revolutionized in some of the solid tumors. And I think in lymphoma, this is going to be a major technique which will change the way we are thinking. Not only will it identify populations with unique antigen expression, but also give you a handle on proximity of different types of cells. So you can look at distance analysis from different cellular components. At the same time, there is another technique that is coming up, which is called 10x genomics, which looks slightly differently. Here you have a H&E slide, and H&E slide on this slide, you capture this slide. You can do some of the immunofluorescent stains on the slide and capture the images. And then you use the slide on which you do the probe hybridization, and you look at RNA profiling you would do on the slide and capture the messages, and then you project it back onto the slide. Here the resolution can be quite high, but would not go to single cell levels. But you can at one go you can profile 18,000 genes. So you can do bulk transcriptomics. May not be at a single level, but you can look at this in an architectural context. This is one of their presentations from the 10x genomics group, where they did a collaboration with our University of Washington. And here is a breast cancer case where they looked at immune cells. So you can ask questions very similar to what we have been asking in the architectural context. But not only are you going to get idea about different immune components like CD4 or CD14 or 68, but you can get the entire gene expression of these cells in an architectural context. So I think these technologies are going to make a huge difference in the way we understand the microenvironment of cancers and lymphomas in particular. The last part of my talk, I want to focus on the host factors that can modulate and result in disparities in microenvironment. The microenvironment of a child can be very different from that of a 80-year-old patient. So age is an important factor. So whatever we discussed has to be taken in the context of age. Microenvironment is not constant. Immune system is not constant. And also there are various other modifying factors. In HIV or any other infection could modify the immune system. Primary immune disorder, secondary immune deficiencies, various autoimmune diseases, nutrition, or are there other factors? All these things one has to take into account. Just to address a few of these questions, my own studies, which I did several years back, here we looked at diffuse large B-cell lymphoma microenvironment, compared those in the post-transplant setting, those with immune competent setting. And you can see that there are far more immune cells in the post-transplant diffuse large B-cell lymphoma, particularly CD8 positive cells were a lot more as compared to immune competent similar type of diffuse large B-cell lymphoma. And if these, in many cases in the post-transplant diffuse large B-cell lymphoma, these T cells were oligoclonal or even clonal. And you can also see some of these cells are slightly atypical, they're highly stimulated cells. And we also looked at other microenvironment components. Here is the analysis on plasmacytoid dendritic cells using CD123 and BDCA2 antibody. And if we looked at three groups, one was post-transplant diffuse large B-cell lymphoma, HIV diffuse large B-cell lymphoma, and immune competent diffuse large B-cell lymphoma. The maximum numbers of plasmacytoid dendritic cells were seen in the post-transplant setting, intermediate numbers in HIV setting, and the least numbers in immune competent setting. So the microenvironment changes with the context, the immune context of the host. And we did a similar study in Hodgkin lymphoma, comparing the microenvironment of Hodgkin's in the HIV positive setting as against the HIV negative setting. And here you can see that there are many more FOXP3 positive cells and CD8 positive cells in the HIV setting, but also it uniquely identifies many CD8 and FOXP3 double positive cells. Here CD8 is in red and FOXP3 in brown, and you can see there are FOXP3 positive CD8 positive cells, whereas we did not see CD8 and FOXP3 double positive cells in the HIV negative setting, Hodgkin lymphoma. Similarly, CD56 NK cells were very few in Hodgkin lymphoma of the HIV positive setting. And similar differences we saw between CD57 positive NKT cells and CD123 positive plasmacytoid dendritic cells being far fewer in HIV positive setting. So this brings into various questions about we can't see microenvironment without knowing the context of the host. In this background, I've been here now for about 10 months in the Fred Hutch, and I started looking at the whole Washington state, how lymphoma patients are doing here. Washington state, as you can see, is a huge state, but a very low population. It's only around 7.7 million population. I think this is the population of Bombay is far more than this. And this is a huge state with 39 counties. And when we looked at the population registry data on incidence and survival, there were some important differences. Seattle is King County is this is King County, and Seattle would come within the King County area. And if you look at the incidence rate of Hodgkin lymphoma, the least incidence rate is in the central region, but you see the mortality rate in central region is the worst, whereas in the King County incidence is fairly high, but mortality is very low, whereas in the northern region and central region, mortality is much higher than in King County. And similarly, when we looked at non-Hodgkin lymphomas, you see that the incidence is low in the central region and the mortality is high in the central region. So there are factors which affect patient outcomes regionally, say even within a state like the Washington state. So the reasons could be multiple. I mean, the reasons could be there's differences in healthcare access or quality. There could be socioeconomic factors, racial distribution can be different between these areas. Environmental agents may be there. I mean, that this is something we feel is there, it's preventable. And all these things may impact on the outcome, or they may impact through the tumor microenvironment. There have been early studies which have come out in breast cancer, where they have shown that race impacts on the microenvironment in breast cancer, which then impacts on patient outcomes. But all these kind of studies which looks at race, one has to be very careful in looking at the data coming out of the impact of race on disparity, because race is not a simple factor, because race also, patients who are racially different may also have other factors. The socioeconomic, they may be disadvantaged, they may be disadvantaged in terms of healthcare access. So this is a very complex field which needs to be assessed, and the tumor microenvironment is one of the key aspects that need to be assessed in terms of disparities in treatment. And in a place like India, this becomes very relevant. Like the same disease can have very differing outcomes in different parts of the country. And I think all aspects, including tumor microenvironment, has to be looked into. I think that's my last slide. Thank you.
Thank you so much, Dr. Naresh. Great lecture. For me, at least, every word was a new learning. I'm sure many people must have benefited tremendously from this wonderful talk. A very different subject and very ably handled. So we have now questions from the audience and the faculty here who would like to interact with you. Maybe we can start with Dr. Sandeep Sheth.
Hello, Dr. Naresh. It was great. Always great to listen talking about lymphoma from you. Very complex interplay of various mechanisms and agents, and very well explained by you. Basically, I work in a tertiary hospital in Ahmedabad, which has a very busy neurosurgical department. And we keep getting a lot of CNS tumors, and every once in a while we get CNS lymphomas. So I just wanted to have your opinion on CNS lymphoma, because they behave very aggressively, and how microenvironment and PD-L1 is going to help or understand the disease process in CNS lymphoma.
Yeah, I mean, primary CNS lymphomas, I haven't really studied from the microenvironment point of view, but I think there are various reasons why they are more aggressive. I think the most important thing being deliverability of drugs. I mean, the what we have traditionally understood as the blood-brain barrier, and that's one of the key issues in treating CNS lymphomas. But I think more work is needed to understand whether microenvironment would play a role in why these lymphomas behave worse, but there could be other reasons. I think it's a bit premature for me to answer that.
They consider, I think, CNS and testicular lymphomas within that immune sanctuary.
Privileged sites, yeah. I mean, the immune privileged sites can work in both ways. Some of the immune privileged sites, they may have like testicular follicular lymphoma is extremely well behaved.
Recording stopped.
Testicular follicular lymphoma behaves in an extremely indolent manner, as against testicular diffuse large B-cell lymphoma.
Yeah.
Testicular large B-cell lymphoma, the most likely recurrence would be in the CNS site, and often give intrathecal methotrexate as a prophylaxis. Whereas testicular follicular lymphoma, which slightly occurs in paratesticular areas, but behaves in a very indolent manner. So these are immune privileged sites, but whether that in itself is responsible, I think it's again an oversimplification of our current understanding.
So PD-L1 has any role in that?
Yeah, I mean, what I would suggest is PD-L1. I tend to use PD-L1 quite a lot in lymphomas for two reasons. One is to in lymphomas outside of especially in mediastinal lymphomas, PD-L1, L2 try to recognize a distinct type of diffuse large B-cell lymphoma, which is a primary mediastinal large B-cell lymphoma. And otherwise is to often the kind of samples that patients we get are cases who have already recurred. So we try to document whether PD-L1 is expressed on lymphoma cells or not, in both in lymphoma cells and in macrophages. And if PD-L1 is expressed, I think it's always important to report that. And independent of whether it is primary, if there is a CNS lymphoma or whether it is a systemic lymphoma elsewhere, I think PD-L1 documentation, I think, is important now.
Thank you.
Thank you, Dr. Sandeep. We take one question from the audience. Dr. Sajin Raj Bhandari wants to know, it's regarding expression of Treg cells in the microenvironment. Is there increased expression in immunogenic tumor the result of an intrinsic host response rather than the tumor cells that have been believed to recruit them?
Expression of what? I didn't follow that.
Treg cells.
Oh, Treg cells. Okay. Now, I think most of the Treg cells are recruited by the lymphoma cells. You know, when lymphoma cells produce cytokines, in the normal physiology, Tregs transition from a T cell response to a CD4 response to a Treg occurs because they have been stimulated long enough. Tregs come into play. But I think lymphoma produces cytokines which will recruit regulatory T cells. I think that's the current thinking.
Thank you. Dr. Tara Roshni Paul. Unmute yourself.
Yeah, good evening. That was a very informative talk on something that we are not too, into. Considering the microenvironment. We practice at a government hospital where resources are limited. And, just getting the diagnosis with lymphoma itself is, we give the diagnosis. So in that background, are there a minimal panel of IHCs to talk about the microenvironment that we could use in different lymphomas, say Hodgkin's or whatever?
Yeah, I don't think it is any different in any other place. I don't think in most places people are not doing adequate work for the microenvironment. Yeah, people may, but what people do but they don't report is like almost everybody does a CD3. Whoever does immunohistochemistry in a lymphoma, they would do CD3. But people don't pay attention to quantify or even semi-quantify the number of T cells. So I think as a first practice, for many years now, I think I've been trying to record, okay, this case has about 20% reactive T cells. So I tend to record that, hoping that someday it's going to be useful. But I think two things, recording the number of T cells, recording the number of macrophages, I think would be good. And I think these things have to be done in the setting of a properly conducted study to get information. So, but if we are using these stains otherwise for classification, at least to record the proportion of cells, I think it's going to be useful. And if you have an interested clinician who keeps record of outcomes of patients and patients are going to be treated in a uniform way, I think it is better to do a little bit of investment into at least the FOXP3 positive cells, cytotoxic T cells, and CD163 to record the amount of macrophages and the infiltration. Again, one has to be very careful. How do we quantify this? A semi-quantitative method of expressing it as a percentage by eyeballing is not easy. We tend to do with CD3, with T cells, you could still do that, but with macrophages, it becomes very difficult because they are much larger cells. So I think we are moving towards sort of more and more centers are using whole slide imaging for routine reporting. I suppose with those things, algorithms will come where you can express these as percentages.
Thanks.
Thanks, Professor Tara. Another question from audience. Dr. Chikodi wants to understand what exactly are ligands.
Ligands are molecules which are released either in the environment or some of them may be expressed on other cells. It can be both on the cell surface or it can be in the stroma, which will bind to a receptor and give a signal. Typical example is PD-L1, PD-L2 binding to PD-1. That's what probably most people would have heard a lot about. And PD-L1, PD-L2 is expressed on the cell surface.
Thank you. Dr. Udayan?
Yeah. Hello, sir. Good evening.
Hello.
Nice to see you. Yeah, I'm fine, sir. Good. Thank you. Well, it has been very tipsy-turvy ride. Actually, in 25 years, we have seen the initial decade where we thought that these are all immune cells which are trying to kill the lymphoma cells. Now we are realizing they're all partners in crime. And you have nicely elucidated that. My question to you, sir, is that what you said and what the literature says is that it is all about B cell lymphomas. For that matter, even Hodgkin lymphoma is a B cell lymphoma. Now, when we are talking everything about T cells and macrophages, why there are no studies or no much literature on the lymphomas belonging to those categories like T cell lymphomas and those derived from the histiocyte macrophage lineage? So, is there any plausible explanation or reason about that?
No, I'm sure there's a lot of work that's going on in the background. I mean, one thing is, like in our own center, we have just started work on mycosis fungoides and Sézary syndrome, looking at the immune cells in the micro. But I think it is far more complex because you also have the complexity of distinguishing which is a neoplastic T cell and which is a reactive T cell. I mean, your problem starts there.
Right.
And it is one thing with all the, on a flow cytometer, probably because of different intensities of antigen expression, you could segregate them as what is neoplastic and what is not neoplastic. And I'm hoping that that is going to be possible on Codex with the intensity of expression. But with the other technologies that we have, because in any T cell lymphoma, you have both neoplastic T cells and benign T cells. And unless you are robustly able to sort that out, you're not going to come out with an answer. Yeah. And also just to, there was a trial which used anti-PD-1 antibody in angiomoblastic T cell lymphoma, but I think that study had to be stopped because it stimulated the angiomoblastic T cell lymphoma, and there were quite a few patients who had augmentation of the symptoms. So T cell lymphoma is a different set. I think there's a lot of studies going on, but I think it's not mature enough to talk about.
Yeah. And sir, are you aware of any such thing going on about the solid tumors? Like there, we still consider inflammatory cells as part of tumor immune response, host immune response towards the tumor.
In solid tumors, there's a lot of work. There is a lot of studies which have happened in breast cancer and colon cancer, looking at different immune subsets. I mean, I have not reviewed them in any great detail, but many of these things actually based on whole image, whole slide imaging actually started off with solid tumors.
Right. Great. Thank you, sir.
Thank you, Udyam. Dr. Abhishek Kakru wants to know, T cell NHL is a problem. Do you think that the genomic information is going to help in development of future drugs?
Yeah, I mean, there is a lot of genomics which is already known on T cell lymphomas. And many of the classifications are based on the genomics. Like we now nodal T cell lymphomas, we segregate them as TFH lymphomas and other T cell lymphomas. And anaplastic large cell lymphomas, we know the subsets of anaplastic large cell lymphoma based on the translocations of ALK, TP63, and DUS22. These are all based on genomics. To whether we will have specific drugs or druggable target, I mean, that's another aspect. I'm sure, I mean, there are several pathways in T cell malignancies which will be amenable for drugs. It's just that doing a T cell lymphoma study is far more difficult because there are fewer. Only about 15% of the lymphomas, of non-Hodgkin lymphomas, are T cell lymphomas. So in any center, it becomes more difficult to carry out a clinical trial for T cell lymphomas as against one of the more common B cell lymphomas.
Thank you. One more question from the audience. Dr. Babita Sodi wants to know, why in nanostring studies, less number of macrophages gave better survival, while in one of the slides, more macrophages gave better prognosis?
No, always the macrophages, the M2 macrophages are pro-tumoral. So more macrophages would mean poor survival. And that has always been the case. I mean, I think I showed you only two studies. The one study was in both of them from Hodgkin lymphoma. The one study which came from Randy Gascoigne's group around 2010, that also showed they used CD68. And those which had high CD68 had a poor survival. And the one from the nanostring, which also used there, they used a macrophage signature, which also showed the higher the signature had poor survival. I don't know whether there was a diffuse large B-cell lymphoma, the follicular lymphoma study, which was immune 1 signature and immune 2 signature. In the immune 1 signature, predominantly the messages were from T cells, but a subset of those messages were from macrophages. And that was in 2002 or 2004. At that time, clarity on different types of macrophages was less. So maybe that's the study which you are referring to. But in Hodgkin lymphoma, most studies have shown, some studies have shown that macrophage content does not distinguish or is not discriminatory. Or they did not, they were not able to replicate the results of British Columbia. But none of them have shown that more macrophages have improved survival in Hodgkin's.
Thank you. Dr. Parimal? Unmute yourself, Parimal.
Good morning, sir. Excellent talk. Sir, my question is, when should we have to ask for the T follicular helper cell associated mutation, such as ROA or IDH2 or TEC2, for further classification of T follicular helper lymphoma? Whether it is useful clinically or whether we just leave it that it's a peripheral T cell lymphoma with T follicular helper phenotype?
No, I think it's very important. I think there are two situations where it is useful. One is, as you said, for subtyping of disease between conventional peripheral treatment of PTCL, NOS, and angioimmunoblastic T-cell lymphoma. Because the disease behavior is different. And also there are many more cases of AITL or TFH lymphomas develop B-cell lymphomas in the background. So I think it's important to distinguish the two. And the other end of the spectrum is cases where you have which is subtle involvement by T-cell lymphomas. You are not sure whether this is just a reactive process or an early angioimmunoblastic T-cell lymphoma. Just to give an example, last week I had a case where the lymph node architecture was perfectly maintained. There were small groups of slightly cells with clear cytoplasm around the vessels. And that was sufficient enough to raise the possibility. And when we did immunostains, again, the case had marginal zone expansion as well, and these clear cells around high endothelial venules. But in itself, it was very hard. So we had to do T-cell clonality studies, and we had to do mutation studies. It was clonal, and there were 2 mutations in TET2 at 3% VAF, and row A was also mutated at 3%. So thereby, it was crucial to give a different definitive diagnosis of angioimmunoblastic T-cell lymphoma, albeit that it is very focally and subtly involving the lymph node, and along with marginal zone of aplasia. And as you know, even in mild lymphomas, you can have TFH expansion. Most often they are reactive. But I think in time to come, you may see papers, which we just sent one paper for publication. You can, the same clone, which is CD4 positive clone giving rise to angioimmunoblastic T-cell lymphoma, can also give rise to a B-cell clone, which can have a phenotype of marginal zone lymphoma. Or sometimes you can have, we had a case where it was full of CD8 positive T cells. And those CD8 positive T cells carried the same mutations as the angioimmunoblastic T-cell lymphoma. And that case initially we called it as a PTCL NOS CD8 phenotype. But when we did all the mutation studies, which was much in retrospect, we did that. And it had all the mutations, and we could microdissect the cells and show that it's carrying the same mutation. So phenotype can change, but to show that it is at least initially arising from an angioimmunoblastic T-cell lymphoma or a TFH cell, you need to do these mutation studies.
And sir, one same thing, that angioimmunoblastic T-cell lymphoma diagnosed case may be relapse as a T-cell lymphoma with a follicular helper phenotype or with follicular lymphoma pattern. So that is whether the AITL and TFH follicular pattern are still the same spectrum of the disease.
Yeah, I think it is the same spectrum of the disease. And I think currently those classifications are still there because those patients who present with follicular T-cell lymphoma, they tend to have a better behavior. But if initially patient presented with an angioblastic T-cell lymphoma, they present later with a more follicular pattern, I would still call it angioblastic T-cell lymphoma.
Thank you, sir.
Thank you, Dr. Parimal. Dr. Ajay Kumar Jha wants to know, this information on tumor microenvironment, has it been clinically utilized?
I don't mean, it has provided a host of targets. So, but where it is, so in that way, I think the microenvironment, like the classic example is PD-1, PD-L inhibitors. So it has been used. But where I think it becomes more crucial is to understand how the microenvironment is different in different populations, in different settings. I think that is really key, because we, like Hodgkin lymphoma, in one area, if 95% of patients get cured of Hodgkin lymphoma, and in another setting, 30% of patients die of Hodgkin lymphoma, we need to understand. And the treatment that we provide for Hodgkin lymphoma may have to change depending on the microenvironment. So I think in a place like India, I think it becomes very relevant to understand the microenvironment and tailor the treatment to microenvironment. See, currently what has happened, we understood, at least with B-cell lymphoma, we have understood a lot about genomics, and genomics is a part of the classification. So if we say something as a germinal center type diffuse large B-cell lymphoma in Bombay or in center in, say, in Trivandrum, we are meaning the same disease. But what we don't know is, are the microenvironment different between a lymphoma of GCB subtype in Bombay, where a GCB DLBCL in Trivandrum, and how that can bring about disparity in outcomes.
Thank you. Before I proceed further, Naresh, can I ask you one question?
Sure.
In the post-transplant lymphoproliferative disorders, rituximab is very effective in controlling the EB virus-induced problems. Now, Hodgkin lymphoma, especially on this part of the world, is all EB virus positive, but rituximab has nothing to do with its treatment. Any thoughts on that?
Yeah, I mean, because most Hodgkins do not express CD20. And even those cases, like 30% cases which express CD20, is expressed in a proportion of cells, and it is very weak. So that's one reason why you don't get a good response in Hodgkin's. There's other thing that I want to mention, like you brought about an important aspect, that post-transplant diffuse large B-cell lymphoma, or PTLD in general, respond remarkably well for minimal treatment, even immune modulation. And when we looked at the mutational spectrum of post-transplant lymphoproliferative disorders and compared them with immune-competent diffuse large B-cell lymphoma, I didn't show that. Almost all the EBV positive PTLDs have a very low mutational load. EBV seems to replace the requirement of many mutations. So either a EBV negative post-transplant diffuse large B-cell lymphoma or an immune-competent EBV negative diffuse large B-cell lymphoma, they will have 2 to 3 times the number of mutations in various genes as compared to a post-transplant lymphoproliferative disorder. So that's the reason why I think with much less treatment, PTLD responds well, especially the EBV positive PTLDs.
Thank you. Dr. Ankur?
Good evening, sir.
Good evening.
It's a very complex topic, and you have explained in a very, very simple manner, sir. Excellent job, sir. So just want to know your experience of the role of myeloid depressor cells in the microenvironment of lymphomas, and your choice of antibodies in form of either flow cytometry or IHC, sir. What is your experience in this, sir?
My experience is zero. In myeloid, that aspect I have not studied. In none of the cases that we studied on Hodgkin's or in non-Hodgkin lymphoma, we looked at the myeloid suppressor cells. And I think it has not been studied as well.
Yes, sir. Because there are very few papers. Yes, sir.
Part of that is because of finding the robust, appropriate antibodies on tissue sections. And till date, I think we were all constrained by the requirement of gene expression. Now that centers outside of those centers which had major, microarray settings can practice many of these things. I think these things, it will open up.
Thank you, sir.
Thank you, Dr. Ankur. Again, a question from Dr. Abhishek. He wants to know your opinion about the circulating tumor cells in lymphomas and their role.
I mean, circulating lymphoma, you think about circulating DNA or circulating lymphoma cells?
It's called it tumor cells.
In some of the lymphomas, you do have circulating cells, and it varies from case to case. More indolent B-cell lymphomas have a higher load of circulating tumor cells. And some of the T-cell lymphomas, like angiomoblastic T-cell lymphoma, also you can see circulating CD4 positive, CD10 positive T cells. So that's a useful, but one has to be very careful in the sense you also have monoclonal lymphocytosis, monoclonal B-cell lymphocytosis. And just because you have a circulating clone, unless and until you can demonstrate that the clone and what you see in peripheral blood is the same as the clone that you have in the lymphoma, it doesn't make it to be the same disease. You can have a completely different MBL in peripheral blood, which is totally different from the what you see in the lymphoma. So one has to be careful. Similarly, when you're looking at marrow, you can have multiple clones of plasma cells, multiple different clones of B cells, and just because there is a clone does not make it a lymphoma or does not make it a myeloma.
Dr. Sindhura.
Thank you, sir. Such a wonderful talk, sir. Very complex topic, but you explained it very nicely, and it was a very excellent review of literature. I had a couple of questions. So recent studies have shown that PD-1 and PD-L1 positive T cells and macrophages in the tumor microenvironment and also increased expression of this LAG3, TIM3 they indicate poor overall survival, and particularly in DLBCL. And while CTLA4 expression in T cells of tumor microenvironment indicate better survival. So what is the cause for such difference? I would have thought that all of them should indicate poor survival.
Yeah, I showed you one paper from a very good group which looked at FOXP3 positive cells, and they got an outcome which was completely contrary to what one would expect. I think much of these things is because one shouldn't look at this in isolation. They have to look at all aspects of the microenvironment and then look at the balancing factors in the microenvironment. That's the strength of the study, say, using nanostring, because they're looking at 90 or 100 transcripts, which gives the entire picture rather than single molecules. So when you look at just CTLA4 without looking at other balancing factors of CD28, so you don't know which way it is going to tilt. So these are all alternate pathways, and I think people have to look at multiple markers which can balance the tumor microenvironment.
I think Codex
the general direction is, if you do a meta-analysis on all of them, I think that would conform to the expected norms. But single studies, there are outliers. If you look at, I showed you one single table which looked at, which has tried to compile all the studies. There are outliers in the studies. I think that's more likely to be deficiencies of individual studies of looking at single molecules.
Sir, I had another question, if I may.
Yep.
Yes, sir. Sir, what is the now because we studied about tumor microenvironment, the CAR T therapy has come into picture, and a lot of advances have been happened. What is the role of tumor microenvironment in cytokine release syndrome that we see in CAR T therapy? So could you please comment on that?
That's a very good question. Currently, we are doing a study on specifically that we are looking at a set of diffuse large B-cell lymphoma patients who responded and a set of diffuse large B-cell lymphoma patients who did not respond to CAR T-cell therapy. And looking at how they, using multiplex immunohistochemistry and codex, looking at the differences in these populations. And I mean, our results are very early, and we are seeing some crucial differences in CD8 positive, in the phenotype of CD8 positive cells, though just looking at the absolute numbers, they look similar. The phenotype of CD8 positive cells are different between the two groups. And the other thing which we have also seen is the pattern of infiltration. There are lymphomas where you see a zone of macrophages and a zone of T cells, and at the right of the center of the tumor, you don't see any T cells. So one thing is to say, what is the total number of CD8 positive cytotoxic T cell content? But in the topography of how the infiltrate is there in the lymphoma cell, if you have a lot of CD8 positive cells at the very periphery, but very few right in the center, that doesn't do any good for the patient, even when you treat patients with CAR T cells. So I think our early thing is that how well these are mixed with each other. What is the proximity of the lymphoma cell to the infiltrate? And that we calculate by doing a proximity analysis to say what is the median distance between the lymphoma cell and a particular immune cell. So even if you have a lot of cells that are clustered somewhere else, but giving rise to a density which is high, but they are no close to the lymphoma cell, it doesn't help. So these are some of the questions we are looking. And we are also looking at the other question which you asked about cytokine release syndrome. That we are doing in a slightly different manner. We are looking at bone marrow samples of nearly longitudinal samples of, in our institute, we have treated now about 400 or 500 cases with CAR T cells. And in many of them, we have got multiple bone marrow samples. So we had a presentation sometime a few months back in the EHP meeting at how the CAR T cells impact not just on the lymphoma cells, but also on bone marrow other cells. So you can get, by day 28, you start getting anemia, you get thrombocytopenia, so you have attack on red cells. Is the red cell precursor is the most common thing that can happen. And beautifully, and also you can see the persistence of T cells for a long time in the bone marrow. The other question, the technical problem that we are facing is how do you distinguish between the regular T cells that have come into the bone marrow and form clusters versus what are CAR T cells? For that, there's a technical problem. We don't have a simple immunostain. You got to do an in situ hybridization to distinguish between CAR T cell and other T cells. So some of the technical problems we are trying to surpass.
Thank you, sir. Thank you.
Thank you. Dr. Sajin has one more question. In solid tumor, CD8 memory cells have been proven to be of good prognostic value when expressed in the tumor microenvironment. Are there expressions seen within the lymphoma microenvironment?
Yeah, in general, that's whether it is lymphoma or solid tumor, having CD8 memory cells, but the memory cells, which are reacting to the tumor antigen, that's always going to be good. But I don't think there have been any studies which have looked at the repertoire of the T cells, because just showing that there are more memory cells doesn't help. They have to have a T cell repertoire against the antigen on the lymphoma cell. We are not there yet.
Again, Naresh, I have one question about lenalidomide, which works through the tumor environment. It is so effective in follicular lymphoma and mantle cell lymphoma, and it is hopeless in DLBCL and Hodgkin lymphoma. Is there any work existing on what's the difference, why it happens?
Lenalidomide, if I remember correctly, correct me if I'm wrong, apart from immune modulation, it also works on angiogenesis, isn't it?
Yeah, sure, it works. But that probably is a minor role and the immune is the major role.
Immune is the major role. Yeah, I don't know the difference, but
Right. Okay.
I mean, many of these drugs, we take them thinking about one mechanism of action, but they have probably multiple mechanisms.
Absolutely. Sure. Dr. Amrita Saran.
Hi everyone. It was a beautiful talk. We really enjoyed it, and you have covered, almost all the aspects of such a complicated topic and really simplified it. So thank you for that. I have a couple of questions. Uh, have you ever studied the difference in the microenvironment in patients with lymphoma with coexistent COVID-19?
I mean, the brief answer to that is no, but I have looked at samples of lymphomas who have had COVID-19, especially follicular lymphomas. We have seen quite a few, and CLLs we have seen because they are more frequent. Nothing that I can say as being very different, but we also looked at some of the lymph nodes which appeared, lymphadenopathy which appeared following COVID vaccination.
Okay.
There we saw slightly disproportionate amount of cases with peculiar follicular hyperplasia with a bit of follicular lysis. Again, we don't have a control arm to see from other vaccinations. So it's all a bit hodgepodge what what data we have and what kind of samples we have. I think something like this, I think we need to collect, an effort has to be made to collect from multiple centers and with good controls. I don't know, I mean, in India, probably you would be having a large number of cases. It would be good to pool this and study.
Maybe it can. I mean, I don't know how practical will it be.
No, in a sense, one would, COVID infection definitely suppresses CD8 T cells.
Right.
And whether that should actually make the lymphoma progress faster. I mean, even if we can't prove that, looking at simple aspects of CD8 density and CD8 expression of other molecules, I think that would be useful, I think. I mean, the COVID, we looked at the microenvironment of various organs in patients with COVID autopsies. We had just around when the time I was living in London, we had, I think, 11 or 12 cases of COVID autopsies where we extensively phenotyped various organs. And it was amazing the way it completely wiped out, suppressed CD8 positive T cells. You could see the entire spleens without hardly any serotoxic T cells. But those were patients who finally succumbed to disease. But patients who have milder symptoms of COVID, it may not be as bad. So, and I couldn't get samples from those patients. So if you have patients of lymphomas with COVID, I think it's worthwhile to study.
Okay, thank you. There is one more question if I'm allowed to ask.
Sure.
Is it possible to reprogram the tumor microenvironment to overcome the resistance to immunotherapy in lymphoma patients?
I mean, I don't know. I'm trying to think of an example, but I mean, the short answer to that is you can reprogram the microenvironment, but the question in the meanwhile is whether the lymphomas have become independent of the microenvironment or have, like I showed you, case of follicular lymphoma, because of some genomic changes, they may shift the dependency from TFH to follicular dendritic cell. So within any lymphoma, there are always subclones which have got some genomic abnormalities, so they are dependent on something else or not dependent on anything. So when you try to manipulate, you also the lymphoma is also clever in the sense that the clone which is independent of the mechanism that you're trying to do may overgrow. But the short answer to that is it should be possible to manipulate the microenvironment and reprogram the microenvironment, provided we understand all the subclones within the population.
Right.
I mean, we understand so much about clonal expansion in the genomic sense. The next challenge will be how do we understand the subclonal complexity in terms of its microenvironment dependence and interactions.
Right. Thank you. There is one last question. Do you think, is there any role of tumor mutational burden in lymphoma as a predictive biomarker?
Yeah, surely. I mean, mutational load, I think, is a very general good surrogate for all the insults that has happened in any cancer. And I think lymphoma is just like any other cancer. The higher the mutational load, the greater diversity, there are greater subclones. And I think the heterogeneity of subclonal population, I think that's the great driver. And in some cases, it is very well studied. And, like in follicular lymphoma and diffuse large B-cell lymphoma, there have been a large number of studies to look at clonal evolution.
Right. Thank you.
Thank you. A question from Dr. Mahesh Mandolkar. Is CD68 positivity in classical Hodgkin lymphoma associated with better prognosis? If so, why is that increased number of CD68 positive macrophages in classical Hodgkin lymphoma is associated with poor prognosis?
Now, I think one has to be careful. The initial study from British Columbia used CD68, but they used a clone of KP1. The KP1 clone stains not only the macrophages, but also neutrophils and eosinophils and everything. So I'm really not sure why they used that clone. There's another clone of CD68, which is the PGM1 clone, which is specific for macrophages and monocytes. I mean, I suspect, the results would have been much cleaner had they used the PGM1 clone than the KP1 clone. But the current thinking is, if there is a lot of M2 macrophages, and which probably is better addressed using CD163, those patients do worse. I think, again, CD68, when you're using, make sure that it is a PGM1 clone and not a KP1 clone.
Thank you. Any other question from the faculty here? Yes, Udaya.
Yeah, sir, I have a question that how do you deal with the issue of tumor heterogeneity while studying from biological perspective, as well as what are your views about can it pose some threat kind of situation in practical utility? Because when we study lymphoma, we see a lot of heterogeneity in a given case or even from one lymph node to the other. Now, I'm sure that must be translating into the difference in the microenvironment in different areas. Now, is there any first-hand experience or your views on that?
I think tumor heterogeneity is very important. We do it routinely. Like, let me take an example. Say you got 5 blocks of follicular lymphoma. You look at every block of follicular lymphoma to identify that one block where you see a diffuse large B-cell lymphoma. So that indicates a heterogeneity in a way that everybody understands, that there is a focal change that occurs, brings about. But the same heterogeneity, you can carry it when you look at biomarkers. If you are using p53 immunostain, having a scattered, strongly positive p53 cell across the whole section may not be that worrisome, as you suddenly see a cluster of, say, 20 cells of strongly positive p53 cell. That is a definite subclone. So that would be alarming.
This is about microenvironment, like in one area of Hodgkin lymphoma. Yeah.
In the microenvironment, as I gave you an example, the best examples I have seen in microenvironment are in diffuse large B-cell lymphoma.
Yeah.
You can see heterogeneity in the infiltrate. You can see cases where the entire lymphoma, the central part of the lymphoma, is what we call as immunogenically cold. That means there are very few immune cells, but all the immune cells are restricted to somewhere in the periphery. And often that area also has fibrosis. Extracellular matrix is pro-tumorigenic, I mentioned to you. So you can see fibrosis, collagen laid down, and on the other side, in those areas, you see a lot of immune cells. And those patients don't respond well because, say, to CAR T-cell treatment, because the immune cells, the CAR T-cells can never reach the center of the where the lymphoma is growing. So,
So do you think that should cause some problem on practical grounds when when it comes to reporting on the cases? Say Dr. Agarwal refers some case to a pathologist who reports that it is PD1 this much percentage, and the other pathologist who has received another section and he reports lesser number of things, and the entire therapy changes.
Yeah, I mean, numerical eyeballing of cells is a difficult thing, not just in the area of immune cells, even with tumor cells. Like, I mean, somebody calls myeloma percentage as 50% and somebody says it is 65%, the treatment can change if the patient doesn't have any other CRAP criteria.
Right.
And are we so perfect in distinguishing between 55% and 65% by eyeballing? No. So this is the percentage count is a bigger problem.
Mm-hmm.
But aspects of geographic heterogeneity is something which is more easily recognizable.
Thank you.
Yeah, Parimal.
Sir, my question is, in peripheral T-cell lymphoma, not otherwise specified, whether you should recommend it to perform the GETA3 and TBX21 routinely by IHC or not, because these two are the different categories of T-cell lymphoma, and that maybe we can suggest the clinician to change the treatment or to add some more drugs.
Yeah, my personal feeling is it should be done, and I tend to do GATA3 in every single case because they are quite often mutually exclusive. TBX21, we have not standardized it well, so I'm not using it in routine work. But GATA3 I always use, and the international community is not uniform in this. There is quite a bit of resistance to say that these categories are not very uniform. Like even if you look at CD4, CD8 phenotypes, it's not distinctly that one is CD4, other is CD8. So that heterogeneity is there. So I'm not even sure whether it will make it to the next WHO classification.
Sir, one more question. That, sir, whether you believe that Lennard lymphoma it still exists or not?
And again, this is a personal belief that there are, within the peripheral T-cell lymphoma, you have a CD8 positive T-cell lymphoma, which is small to medium size, with very few, other cells, a tumor cell rich type. And that, I think that is quite unique. And that's what I would say as a Lennard's lymphoma with epithelioid cells. But bulk of the T cells are medium sized and little more monomorphic as compared to other peripheral T cell lymphomas. And I think that does exist. But again, these are so rare. Like in a year, I may see 2 cases of what I would call Lennard's lymphoma. So it's hard to have very convictions on these things.
Thank you. Dr. Sandeep.
My question is very general. With so much studies and research going on in TME, what do you see the future holds for treatment, for lymphoma treatment?
But I'm hoping with the coming in of Codex and with the 10x genomics and also nanostring, I think it's going to become more, there'll be more and more studies. And clinical trials, I'm sure people will be looking for opportunities to maximize, outcome benefits using this kind of information. At least in a clinical trial setting, I think this in the next 10 years, more and more of TME-based decisions will be made in the clinical trial setting. But it'll probably take another 10 years for it to come into routine practice. Dr. Agarwal, I think you should probably, I think we would like to hear from you on this.
What should I say? What you mentioned about the PD1, PDL1, I mentioned about lenalidomide, and now this macrophage CD47 antibody. These are the probably three glorious examples of how microenvironment has been exploited to treat malignancies, especially as you mentioned, the medicinal large B-cell lymphoma, Hodgkin lymphoma, and then polyneuridomide, follicular lymphoma, mantle cell lymphoma, and where this macrophage antibody business will take us, we don't know. But these are some of the excellent examples of exploiting the microenvironment knowledge. And this may be just the beginning because learning.
Yeah, I think Dr. Agarwal's response brings out a key aspect, like the study of microenvironment has to be done it has to be a team effort. Like the understanding about drugs comes from a different kind of expertise. The understanding about identifying the druggable targets come from a different kind of expertise of people, of hardcore molecular biologists, of identifying the who can do studies of algorithms about how the molecule changes, and then you need people like us, hematopathologists, who can produce the right kind of platforms to ask these questions and look at how, in the concept of our understanding of pathology and lymphomas, we can fit these principles into the pathology background. So it requires people of multiple backgrounds to come together to really get good meaning out of this. And no single person will be able to have knowledge and really critical thinking of all these aspects. One thing is up to reading and having knowledge, but having to be able to critically analyze, I think you require a group for this. It's not like lymphoma classification, which is different.
Parimal, yes.
Sir, one question related to immunohistochemistry, that in high-grade B-cell lymphoma with TDT positive, so whether in how many TDT positive cells should be there to differentiate high-grade B-cell lymphoma TDT positive from B lymphoblastic lymphoma, whether the percentage of TDT positive as well as whether the intensity of TDT will help to differentiate both these entities?
I think TDT positivity should be taken as only one aspect. Of course, percentage is important. If like more than 50% cells are positive, you would think that they're lymphoblastic. But other aspects also one has to see, whether it's expressing surface immunoglobulin or whether it's only cytoplasmic immunoglobulin. There is a nice scoring system which was published maybe a few months back from the MD Anderson group, looking at multiple aspects about CD20 expression, TDT expression, surface immunoglobulin expression, and a few other things. I think it's worthwhile to look at it. And also MYC expression. Most of the double hit lymphomas, which are, MYC and BCL2 positive, tend to have TDT expression. So understanding it in the context of the genomic abnormalities is also important.
Thanks.
Thank you, Parimal. One last question from the audience. In heavily pretreated lymphomas, the TME might be different than in a newly diagnosed case. With regards to ICB, should they be offered much earlier as treatment options in the disease course, as the potency of the cytotoxic T cells could be less due to immunosuppressive therapies?
Yeah, probably I think you're best to answer this question. Obviously, post-treatment, heavily treated cases, their microenvironment is heavily manipulated, no doubt. But the practicalities of doing something novel on a patient who is not as a first-line treatment, and the difficulties of using something experimental as a first-line treatment. Probably I will leave it to you to answer the question.
It's extremely difficult. You are absolutely right. Any other questions? We just have a minute or two. If no, then Dr. Naresh, thank you so much. Sparing your valuable time, giving us a wonderful lecture, very different insight, at least for a clinician like me. Maybe the audience here and the faculty here was at home, but for me, it was an experience and a great experience. Thank you so much for sharing all this with us. Thanks to the faculty.
Thank you.
Thank you.
Thank you.
Thank you. Thank you.
Thank you, sir.
Naresh sir. Thank you, Dr. Agarwal.
Thank you very much, sir.
Thank you
And
so
you
much.
too,
Have a
sir.
nice evening.
Thanks to the audience. Thanks
Giving
to
us the opportunity. Thank you.
and thanks to the sponsors RBG. Thank you everybody. And Naresh, we will keep bothering you.
No problem. Pleasure.
Thank you. Bye. Enjoy.
Bye-bye.
Bye.
And good night to all the Indians.
Have a good day.
Right, sir.